Welcome to my video lecture. وياكم دكتورة شيماء. So today's topic is enzyme-linked immunosorbent assay, or it's called ELISA. Before we go through our lecture, please answer the question in the classwork, and you are allowed to answer the question during the time of the lecture. So خلال فترة وقت المحاضرة أني أستلم الإجابات لأنه يعتبر حضور إلكم. Enzyme-linked immunosorbent assay, or the ELISA technique, is a colorimetric test. It is a qualitative and quantitative immunological procedure in which the antigen-antibody reaction is monitored by enzyme measurement, and the ELISA reaction are set up in 96 well plate or other size of the plate, depend on how much sample you have. And these plates are read by a plate reader, which is basically measure the absorbance of a at a specific wavelength, and the result is displayed on a computer screen. And the result is basically the absorbance versus the concentration. From that we understand how much of the substance of the sample is present. It is a diagnostic tool for many viral diseases, example HIV, AIDS, and it's used to quantify substance such as peptide, protein, antibodies, and hormone. The basic step in ELISA technique includes immobilization of the antigen to a solid surface. يعني أقصد بالcoating of the antigen على solid surface اللي هي البليت اللي هو 96 well plate. أكو بليتات بعد أكبر بها وز أكثر. Also, the second step is the complexation of the antigen with the specific antibody اللي هو primary antibodies. يصير عندنا binding of the antigen with the first antibody اللي هو the primary antibody. Then attachment of the primary antibody to the secondary antibody, بس هذا secondary antibody is linked with an enzyme. إذن هو enzyme linked secondary antibodies. Also the next step is the reaction of the substrate with conjugated enzyme to produce a colored product. So when there is an antigen binding with the primary antibody, then the secondary antibody that is being linked with an enzyme. So if this reaction happens, and when we add the substrate and there is a correct binding of the antigen with the antibodies, the color will develop, and that's what the plate reader will detect. لأنه هي قلنا this technique is a colorimetric test يعتمد على development of color. إذا صار عندي antigen antibody binding, ال substrate which we add will cause a change of the color of the enzyme which is linked to the secondary antibodies. فتغير اللون هو دلالة على وجود binding of antigen with the antibody. ELISA is a popular format of wet lab type assay that uses a solid phase. Solid phase هو أقصد به microtiter plate أو 96 well plate. Enzyme immunoassay to detect the presence of a substance, usually an antigen, in a liquid sample or a wet sample. It involves detection of an analyte, the specific substance whose presence is being quantitatively and qualitatively analyzed. So احنا قلنا from the beginning it is a qualitative and a quantitative technique. The analyte is also called the ligand, and it will specifically bind to a detection antigen. As we said, the ligand specific binding reagent is immobilized or coated on a stationary solid phase اللي هي الإليزا بليت أو اللي هي المايكروتايتر بليت أو قد تكون 96 wall plate مثل ما قلنا. It is then complex with an antibody that is linked to an enzyme. Detection is accomplished by assessing the conjugated enzyme activity by incubation with a substrate. يعني substrate هو إذا شاف أكو binding antigen antibody binding, so هو اللي يغير ال color of the enzyme which is linked to the antibody from colorless into a blue color, which form a colored product. The most crucial element of detection strategy is highly specific antigen antibody interaction. هذه الخطوات أو البروتوكول اللي يتبع بالدايركت إليزا تكنيك. أول خطوة هي عندنا الكوتينج of the antigen. طبعا الانتيجين يكون soluble in buffer معين، فنضيف الانتيجين and we leave it depending on the instruction of the kit. إذا كان نستخدم كit جاهز قسم ينصحون ب 1 hour incubation at room temperature. On some experiment it is advised to incubate it overnight. Then after the incubation period is finished, we will remove the buffer and wash the plate. طبعا هم نستخدم بفرات معينة للwashing، ممكن نسوي له washing 2 times or 3 times. Then نجي على خطوة step of blocking. نضيف blocking buffer. هذا will block the free site in the wells حتى من نضيف احنا الانتيبوديز من نضيفها ما يصير binding على other antigen or anything in the well, so it will bind specifically to the antigen that we have coated. So this is the reason why we add a blocking buffer. And then we will remove the buffer after a certain time of incubation and wash the plate. Then the third step is detection. We add the antibodies that is linked to the enzyme. So enzyme conjugated detection antibodies bind to the antigen. So these enzyme linked antibody will bind to the antigen. Then after we incubate it for a certain time, then we remove the buffer and wash the plate again. Always there is washing after each step, there is a washing. Then we read out by the final step is we add the substrate. So when we add the substrate, if there is antigen antibody binding, so the color will develop. Substrate, as it says here, is catalyzed by an enzyme to generate a colored readout or colored product. As we have seen in the previous schematic diagram, the ELISA begins with a coating step where the first layer, either an antigen or antibody, is adsorbed to the wells in ELISA plate. So either the plate is coated with antigen or antibodies. Coating is followed by a blocking and detection step known as shown in the simple schematic diagram. Since the assay uses surface binding for separation, several washes are repeated between each ELISA step to remove unbound material. So عملية الwashing بين step وأخرى كلش مهمة to remove the unbound material. During this process, it is also essential that excess liquid is removed in order to prevent the dilution of the solution added in the next stage. So احنا من نسوي washing, after washing we have to dry the plate. شلون نسوي له drying البليت؟ مو نحطه بالاوفن مثلا to dry it. بس مجرد نحط كنينس ونضرب البليت على الكنينس 3 مرات فينشف البليت. The most complex and varying step in the overall process is detection, where multiple layers of antibodies can be used to amplify signals. يعني ممكن نستخدم primary antibodies، بعدين نستخدم secondary antibodies، ممكن نستخدم antibodies آخر. هاي can amplify السيجنالز ويعطينا can detect a very small amount of analyte which is present or coated on the plate. And this schematic diagram, we are showing that the walls of the plate can be coated with antibodies. As you see, these Y-shaped protein, these are antibodies coated on the walls of the plate. First, they incubated overnight or 1 hour, depending on the protocol we follow. Then we do a washing step. Then we add the sample containing the antigen and incubate it for a certain time, like 1 hour or 2 hours. Then we do a washing step and we add the primary antibodies, and we do an incubation step. So these antibodies are specifically bind to these antigen. After that, we do a washing step and add the secondary antibody, which is enzyme linked. اللي هو mostly they use the HRP اللي هو Harris Reddish peroxidase. So this is the antibody that is coated on the plate, and this is the sample containing the antigen, and this is the primary antibody, and this is the secondary antibody which is enzyme linked اللي هو ال HRP اللي هو Harris Reddish peroxidase. Then we do a washing step after the incubation period is finished. Then we add the substrate. هسه نشرح أنواع السبستريت اللي ممكن نستخدمها. So if they detect an antigen antibody binding, the color will develop after a short incubation period. Then we measure this plate using a plate reader. ومعظم الإلايزا تكنيك نستخدم ال 450 نانومتر اللي هو الويف length to measure the absorbance. So what do we need for doing an ELISA experiment? We need an ELISA plate. قلنا اكو انواع، اكو 96 wall plate، اكو اكثر اكو اقل، يعتمد على size of the sample. We need buffers, antibodies, enzyme, and we should know how to handle our sample and prepare it. And we need a substrate. طبعا الإلازا البليت اللي تستخدم لها فد مواصفات معينة اللي نستخدمها بالإلازا تكنيك. يجب أن تكون flat bottom because some of the plates have a round bottom or V-shaped bottom. فاللي نستخدمها to coat الانتيجين or the antibody, they are flat bottom. 96 well plate made from polystyrene or polyvinyl chloride are coated with either inactivated antigen or antibodies. طبعا تكون مصنوعة قلنا أما من مادة البوليسترين or polyvinyl chloride اللي حتى يصير به absorption أكثر when we coat with antigen or antibodies. The coating acts as the binding site for the antigen antibody present in the sample. Further variants are the 384 wells, and as we said, there is larger plate depending on the size of the sample. We need different type of buffers when doing an ELISA technique. One for coating, another for blocking. When we said before adding an antibody, we need to block any site so that the antibody will bind specifically to the antigen. Another for washing, we said between each steps. After each step, there is a washing step. and perhaps another one for sample and antibody dilutions. Buffers can be produced in-house. يعني in-house نقصد باللاب، يعني احنا نصنعها واحنا نحضر الدايلوشن مالتها باللاب مالتنا نشتغل بيه، or sourced from a variety of commercial antibodies and reagent supplier، أو نشتريها commercially. but إن شاء الله من إذا صار عندكم فرصة تكملون بالخارج الدراسات حتشوفون إنه معظم اللابات بالخارج هم اللي يحضرون البفرات بإيدهم. قليل ما يشترون لأنه they are very expensive and it is very simple and easy to make, so they make their own buffers. So the coating buffer is the process where a suitably diluted antigen or antibodies is incubated until adsorbed to the surface of the wells. يعني احنا شنو نريد نسوي coating with الانتيجين or antibodies، هذا يصير لازم نسوي له dilution بالcoating buffer حتى to be adsorbed to the plastic plate. Adsorption occurs passively as the result of the hydrophobic interaction between the amino acid side chain on the antibodies or the antigen used for coating and the plastic surface. إذن هو حيصير عندي hydrophobic interaction between الانتيبودي or الانتيجين، يعتمد احنا شنو هنسوي coating على على البليت with the plastic surface of the plate. It is dependent upon time, so adsorption, how strong the adsorption of the antigen or the antibodies on the plastic plate يعتمد على التايم. يعني sometimes 1 hour is not enough, so نسوي troubleshooting، يعني شلون احنا نصلح الاكسبيرمنت مالتنا، نعرف وين خطأنا، نطول الانكوبيشن تايم حتى يصير عندي الانتيجين or the antibody is absorbed very well to the plastic surface of the plate. أيضا يعتمد على temperature. Some experiment it is fine with 4 centigrade، يعني نحطها بالفريج. Other it is fine at 37 at room temperature. And also يعتمد على ال pH, the coating buffer, as well as the concentration of the coating agent. أيضا sometimes إذا نستخدم very low concentration ما حيصير عندي binding very good binding of the antigen with the antibody. فدائما احنا نشتغل experiment لازم نسوي trouble shooting، يعني نعرف احنا وين الخلل. ليش مثلا احنا نعرف هذا الانتيجين will bind very good with the antibody but بالاكسبيرمنت من جينا اشتغلناها ما صار development للcolor. فشنو الخلل؟ ممكن انه الانتيجين التركيز الانتيجين اللي سويت له كوتنج على البليت قد يكون قليل. فكل اكسبيرمنت تحتاج الى شلون trouble shooting حتى تمشي الاكسبيرمنت مالتنا صحيحة. هاي خصوصا اذا بالريسيرش، بس اذا يعني كنت جاهز تستخدمه هو they recommend how much يعني او هم already ما يعني الكتات الجاهزة يكون already the plate is coated with antigen or the antibody، انت بس تضيف السامبل اللي هو يحتوي نوعا على الانتيجين or the antibody. يعتمد which type of ELISA technique is using, the direct or the indirect، هسه نشرحها بالسلايدات القادمة. طبعا إذا كنا مستخدمين بالإلايزا experiment مالتنا ال 96 well plate، each well بالبليت maximum volume اللي نقدر نحطه بالويل هو 200 microliter. فالأفضل احنا نضيف 100 microliter of coating buffer containing 1 إلى 10 microgram per ml of the antigen or the antibody. طبعا الانكوبيشن يعني maybe an overnight أو قلنا 1 إلى 3 hours، يعتمد على البروتوكول. طبعا وين نسوي له incubation؟ قسم experiment is fine doing it at room temperature، يعني leave outside أو نحطه بالكابينت أو نحطها some experiment they work better when the incubation is at 4 centigrade يعني بالفريج. Coating buffer stabilize the antigen or antibody which is used to coat the ELISA multi-well plate, maximizing adsorption. So احنا ليش نسوي dilution للانتيجين or the antibodies in coating buffer is to maximize the adsorption to the plate and optimizing interaction with the detection antibodies لحتى يكون better also binding not only with the plate but with the detection antibodies اللي احنا حنضيفها. The two most common coating buffer are bicarbonate buffer at pH 9.6 or PBS اللي هو phosphate buffer saline. So after coating the plate we need to add a blocking buffer. So to prevent the non-specific binding of detection antibody, there are two main types of blocking agent, protein and detergent. Protein permanent blocking agent and hence added after the capture antibodies has adsorbed to the well surface, and detergent block temporarily so it is not long lasting. Blocking function disappears during washing step. The most basic blocking buffers or the type we use is 1% bovine serum albumin اللي هو 1% BSA or milk protein هو الحليب الباودر dissolved in PBS اللي هو phosphate buffer saline. Usually we add 150 microliter of blocking buffer is added to the wells to incubated for 1 hour at 37 centigrade in order to fully block the plate. So why we do washing with washing buffer? To remove the unbound material. Usually we use PBS, phosphate buffer saline, with a small concentration of a non-ionic detergent such as twin 20 is used. Washing is typically repeated 3 to 5 times between each step in the ELISA to thoroughly remove the unbounded material. Excess wash solution must be removed in the final wash step. يعني شلون اوخر this washing buffer؟ قلنا نقلب البليت على ورق من الكلينس ونحاول انه نضرب البليت فد 3 مرات 4 مرات حتى تروح من الاكسس washing fluid. That will prevent the dilutions of the reagent added in the subsequent stage. لأنه أنا إذا كان عندي موجود washing buffer ما تخلصت منه هذا حيضيف إلى ويغير لي بالconcentration اللي أنا مثلا of the primary antibodies اللي حضيفه بالخطوة اللي قادمة. The antibodies used in ELISA assay can be monoclonal, polyclonal or combination of both. شنو الفرق بين المونوكلونال and polyclonal؟ المونوكلونال antibodies can recognize a single antigen a single epitopes on the antigen. نعرف الانتيجين بها different epitopes. المونوكلونال antibody can recognize a single or unique type of epitopes on the antigen, whereas the polyclonal antibodies can detect different type of epitopes on the same antigen. So the monoclonal antibodies are more specific because they recognize a single epitopes on the antigen. The interaction between antibodies and their antigen is described in 3 ways: specificity, affinity, and avidity. Specificity is an indication of whether an antibody binds slowly to a unique antigen. So we said that the monoclonal antibody are more specific because they can recognize a unique type of epitopes on the antigen. Affinity is described the strength of binding of an antibody to a single antigen. ممكن ان يصير عندي binding between an antigen and the antibodies, but when we نسوي antibody titer نشوف انه بالconcentration الكلش عالي من الانتيبودي يلا دا يصير binding ويا الانتيجين, so they don't have a very high affinity between the antigen and the antibody. There is no strong binding between them. Avidities account for the total stability of the antigen-antibody interaction. So as we mentioned in the previous slide, the monoclonal antibody can be used for all antibodies containing steps in all types of ELISA technique. Monoclonal antibodies are homogeneous by definition with a specificity for a single epitope or small region of the protein, single epitopes on the antigen. Polyclonal antibodies are complex antibodies pools which represent a collection of specificities to various epitopes found in a single antigen. They can also be used as a capture and detection antibodies. يعني ممكن نستخدمها secondary antibodies that is enzyme linked. ولما نضيف substrate they can develop a color if there is an antigen antibody binding. So the most commonly used enzyme label are the horseradish peroxidase HRP and the alkaline phosphatase. هي هذول أكثر الأنواع اللي شائع استخدامها كإنزيم ليبل على الانتيبوديز. فمن نضيف substrate they will develop a color if there is a binding with an antigen. Also there is other type which is less commonly used. So the specimen sample for ELISA includes serum, CSF, cerebrospinal fluid, sputum, urine, Siemens, supernatant of culture, a stool. In the simplest case, ELISA sample are diluted either in PBS اللي هو phosphate buffer saline، ممكن بالwash buffer، أيضا ممكن بالcoating buffer اللي هو sodium bicarb. And these applied in a final volume of 100 microliter. مثل ما ذكرنا انه الدايلوشن مال السامبل ممكن نستخدم 1 الى 10 مايكروغرام per ml بهذه الفاينل فوليوم بالويلز اللي نضيفه هو 100 microliter. A large selection of substrate is available for performing the ELISA with an HRB اللي هو horseradish peroxidase or the alkaline phosphatase conjugate. So the choice of the substrate depends upon the required assay sensitivity and the instrumentation available for signal detection اللي هو spectrophotometer or هو الplate reader. فممكن الplate reader يحتوي لنا على فلاتر to detect the HRB but ما عنده filter to detect the alkaline phosphatase فيعتمد على الانسترومنت اللي احنا موجودة باللاب مالتنا. So we will discuss one by one each of the 4 type of the ELISA technique اللي هو direct ELISA, indirect ELISA, sandwich ELISA and the competition inhibition ELISA type. In the direct ELISA technique, an antigen is immobilized or coated on the wells of an ELISA plate as shown here. Then these antigen is detected by the enzyme linked antibodies, and these enzymes such as horseradish peroxidase or alkaline phosphatase. Then if we add the substrate which detect the antigen antibody binding, the color will be developed. And the advantage of this technique, it will be a very fast technique and less prone to error. يعني أقل نسبة حدوث خطأ لأنه أقل reagent وأقل steps بهذا type of ELISA technique. لكن ممكن يكون disadvantage إنه ما عندي amplification للsignals لأنه we are not using a secondary antibody to amplify the signal. Indirect ELISA technique. In this situation, the wells of the microtiter plates are coated with a known antigen. Here, the antigen are marked or colored in green, then the antibody to be tested is added against the antigen, and hopefully that if the antibodies is specific against this antigen, it would bind to that antigen. Now the second step is adding of the enzyme-linked secondary antibody, and then when adding the substrate, the color is developed. That is an indirect proof that the sample that we provided has the specific antibody against the antigen. So, like for example, if the patient is suspected to have an HIV infection, so we coat the plate with the antigen of this virus, then we add the serum of the patient. Usually, when there is infection with HIV, the immune response will develop antibodies. So the blood of the patient will have antibodies. So we add these serum containing these antibodies, and then we add the secondary antibodies, which is enzyme linked. Then the color develops when adding the substrate. That means the patient is HIV positive. Whereas if the serum does not have these antibodies, the color will not develop, and this confirms that the patient is HIV negative.
هنا حنشوف فيديو على الاندايركت إليزا تكنيك. نعرف هو firstly is the coating of the antigen, ونضيف بعدين ال primary and secondary antibody which is enzyme linked, بعدين نضيف لل substrate. So in the first step هنا هذا ال researcher is preparing the antigen which is diluted 1 microgram per ml in coating buffer اللي هو 0.1 molar carbonate. ممكن نستخدم 1 إلى 10 microgram per ml concentration of the antigen prepared. فدي يسوي له هنا vortex للسامبل. بعدين يسحب about 100 microliter and coating the antigen to the micro titer plate. ممكن هنا نسوي duplicate أو triplicate of number of wells. بعدين نسوي له incubation at room temperature for 3 hours or 4 centigrade overnight. يعتمد إنه إذا مستعجلين على experiment أو عندنا experiment ثانية ممكن نخليه بالفريج overnight. بعدين عندنا remove the excess and then washing step. They wash the plate by filling the wells with 200 microliter 0.2% PBST, يعني phosphate buffer saline with between 20. Then blocking the remaining protein binding site by adding 200 microliter of blocking buffer اللي هو 5% of skim milk, يعني الحليب الباودر in 0.2% PBS. And then incubation step at room temperature for 1 hour or 4 centigrade overnight. Then we remove the blocking buffer. بعدين نسوي له washing the plate twice with PBST, يعني phosphate buffer saline 2020. ونسوي له drying بهذه الطريقة. Then we add 100 microliter of diluted primary antibodies. طبعا ممكن يكون عندي was the control عندي positive control negative control. يعني عندي was ما بيها not coated with antigen ونضيف بس ال primary with secondary antibodies، أو بالعكس بيها antigen وما أضيف ال primary antibodies. These used as a control. So then here the incubation at room temperature and removing the solution after finishing the incubation time, and then washing again 3 times with PBS 2020 and drying step. Then we add 100 microliter of the conjugated secondary antibody diluted at the optimal concentration. يعني concentration يختلف من يعني إذا نستخدم ويسترن بلوت يختلف الدايلوشن عن مثلا إلايزا تكنيك، فهذا يجي ويا الليفلت مال الانزيم لينك secondary antibody. So هم نسوي له incubation for 1 hour at room temperature. خلصت incubation time نسوي له remove لل secondary antibodies and washing step with PBS 2020, and then the drying step حتى لأنه إذا بقى عندي سائل ممكن يأثر لي على الدايلوشن of the next step، حيكون تركيزه مخفف مثلا إذا هسه ضفنا substrate إذا كان باقي من ال solution. Then we dispense 100 microliter of the substrate ويشوف الcolor here develop، وهذه هي الplate reader نقراها على wavelength 450 نانومتر، حيعطينا هذه القراءات اللي هي الابزوربنس. And the sandwich ELISA technique is used to detect antigen of interest. We can coat the walls of the microtiter plate with the antibodies as shown here. These are the captured antibodies are coated on the walls of the microtiter plate. Then we can give the patient serum or the patient's blood. Then we can give another antibodies that is coupled with an enzyme. These are enzyme-linked antibodies. And then we put the substrate. Now, if the monoclonal antibodies coupled with an enzyme is binding to that particular antigen, then in a color, the color will develop. So when the color develops, this it confirmed that the antigen is present in the patient sample. So this is in the direct ELISA. We are using one detecting antibodies. We can use indirect ELISA here by using not only one antibodies. So so here the antibodies are the capture antibodies on the 96 well plate, and these are the antigen which is in the patient serum or blood. Then we add the primary antibodies which is not linked to an enzyme. Then we add the secondary antibodies that is linked to the enzyme. That means it is detected not by two antibodies, it is detected by so there is 3 antibodies involved here. So this technique is very sensitive and specific, and the signal will be amplified. هذا النوع الأخير من الإلايزا تكنيك اللي هو الكومبيتيشن أو انهبيشن تكنيك، هو بس مطلوب منكم بس الاطلاع. هو very complex technique. It is used to measure the antibodies or the antigen concentration in the sample. ويعتمد على the competition of the antigen or the antibodies with a reference binding to a limited amount of labeled antibody or antigen. So these are the 3 type of ELISA technique المطلوبة من عندكم اللي هي الدايركت والاندايركت والساندويتش ELISA technique. بالدايركت قلنا الانتيجين coated on the plate, and then we add the antibodies which is enzymely linked, and when we add the substrate, and if the color develop, this color will be detected by the plate reader. In the indirect ELISA technique, we have also the antigen coated on the plate. Then we add the primary antibody, which is not enzymely linked, and then we add the secondary antibodies conjugate that is enzyme linked, اللي هو الهورس radish peroxidase, for example. And then when we add the substrate, and if the color develop, that color will be detected again by the plate reader. While in the sandwich ELISA technique, then a captured antibody will be coated on the plate. Then we add the serum or the blood that may contain an antigen that we we want to detect. Then we add the primary and the secondary antibody that is enzymely linked, and finally we add the substrate. And this sandwich technique can also be done also by adding a primary antibody that have an enzyme linked and not need necessary to add a secondary antibody. So if this detector antibody could have an enzyme linked, then we can add the substrate, and again, if the color develop, it will be detected by the plate reader.
Application of ELISA technique. It is used for determining the serum antibody concentration, antibody screening as a diagnostic test for many of the viral infections such as HIV, West Nile virus, NDB for home pregnancy tests, and it's also used in food industry. And as we said, in disease diagnosis like HIV, bird flu, common cold, corona, detection of enterotoxins of E. coli in feces, and to study the drug toxicity. Thank you for your attention.